Let's learn about one of the most important things in the universe, DNA and RNA. DNA and RNA are nucleic acids that contain the genetic information which codes for proteins necessary for every living thing to function. Let's begin with the overall structure of DNA. DNA is a double helix composed of two strands of nucleotides that run anti-parallel to each other. The two strands are connected to one another by two types of complementary base-paired nitrogenous bases, a purine and a pyrimidine. When these two strands are coiled, they create the characteristic double helix shape and form two grooves, a major groove, which is the gap between the two coils, and a minor groove between the backbones where the base pairs are. RNA is usually a single-stranded molecule, but it can form unique shapes by base pairing and folding in on itself. RNA is also composed of nucleotides, just a different type than those found in DNA. Now let's look closer at the individual components of DNA. The nucleotides of DNA are called deoxyribonucleotides. They contain a pentose 5-carbon sugar with a phosphate group on the 5' carbon, a hydroxyl group on the 3' carbon, and a nitrogenous base on the 1' carbon. The nucleotides of RNA are called ribonucleotides. They are very similar to deoxyribonucleotides. They also contain a pentose sugar with a 5' phosphate group, a 3' hydroxyl group, and a nitrogenous base on the 1' carbon. But they differ from deoxyribonucleotides in that they have an additional hydroxyl group on the 2' carbon. This is why the DNA nucleotides are called deoxyribonucleotides. They have been deoxized on the 2' carbon. DNA and RNA also differ in the nitrogenous bases they contain. DNA contains the nitrogenous bases adenine, guanine, cytosine, and thymine. RNA contains adenine, guanine, cytosine, and uracil. The nitrogenous bases can be divided into two groups, purines, which are larger two-ringed molecules, and pyrimidines, which are smaller single-ring molecules. The nitrogenous bases have a strict system for how they pair up. Adenine pairs with thymine, or uracil if in RNA, and guanine pairs with cytosine. The nitrogenous bases are bonded together by hydrogen bonds. Adenine and thymine are bonded by 2 hydrogen bonds, while guanine and cytosine are bonded by 3. The 5' phosphate group of one nucleotide covalently bonds to the 3' hydroxyl group of the adjacent nucleotide, forming the backbone of DNA and RNA and explaining why the strands run in the 5' to 3' direction. It is important to learn about the structure of DNA and RNA because structure dictates function. The next important concept we will learn about is DNA replication. In order for genetic information to be passed on to future generations, it must first be replicated. DNA replication in prokaryotes is different from DNA replication in eukaryotes because prokaryotes have circular DNA instead of linear. Let's look at DNA replication in E. coli, for example. To initiate replication, we must first locate the origin of replication, or OREC. This is a non-structural gene that is an A to T rich area. Recall that A-T pairings are held together by 2 hydrogen bonds, which are easier to open up than the 3 hydrogen bonds between G and C. Once the OREC has been located, special DNA A proteins bind to this area and force the DNA to open up. This forms what's called a replication bubble. Each replication bubble contains 2 replication forks. Next, single-stranded binding proteins will bind to the separated DNA strands in order to protect them from degradation. At the replication fork, DNA C protein helps guide DNA B protein in place. DNA B is a helicase enzyme and serves to unwind the DNA at the replication fork so that other enzymes can enter and begin replication. Because the DNA is being unwound, it can cause supercoiling in other areas of the strand and lead to breakage. To alleviate this, a topoisomerase called DNA gyrase binds ahead of the replication forks and counteracts the supercoiling. Once the DNA is unwound, the nucleotides can now be replicated into a new strand of DNA. Recall that the DNA runs in a 5' to 3' direction, and thus new DNA can only be synthesized in this direction. To make things a little easier to understand, let's look at one replication fork at a time. New nucleotides are added to the free 3' hydroxyl group. An enzyme called DNA polymerase III can perform this function, but it can only add nucleotides to an already existing 3' end. A different enzyme called primase is needed. Primase will synthesize what is called an RNA primer that is about 10 nucleotides long. Now that there is an existing 3' end, DNA polymerase can begin adding nucleotides and synthesizing DNA. Because nucleotides can only be synthesized in the 5' to 3' direction, and because there are two antiparallel strands in the replication fork, the 5' to 3' strand will be synthesized continuously, and the 3' to 5' strand will be synthesized discontinuously. The continuous strand is known as the leading strand, and the discontinuous strand is known as the lagging strand. The lagging strand is synthesized with Okazaki fragments, each of which begin with an RNA primer and end with DNA synthesized by DNA polymerase. To convert the fragments into a continuous strand, the enzyme RNase removes the RNA primer of each fragment. And now, DNA polymerase follows behind from the next Okazaki fragment to fill in where the primer was. This still leaves a nick between each fragment, which is then sealed by DNA ligase. Zooming back out to look at the entire replication bubble, we see that there are two continuous strands and two discontinuous strands being synthesized towards each replication fork. Although the discontinuous strands may appear to be synthesized away from the replication fork, the Okazaki fragments are indeed synthesized towards the replication fork. The fragment labeled A is synthesized first, followed by the B fragment, which is synthesized next. Replication continues around the circle until the replication forks meet and all DNA has been copied. Because the new double-stranded DNA molecule contains an original strand and a newly synthesized strand, this type of replication is called semi-conservative replication. Now let's test your knowledge. Complete the following quiz to see what you have learned. Question 1. Identify the correctly drawn deoxyribonucleotide. Question 2. Identify the correctly drawn ribonucleotide. Question 3. Sort the following nitrogenous bases into their correct category. Question 4. Which nitrogenous base is unique to DNA? Question 5. Which nitrogenous base is unique to RNA? Question 6. Which of the following correctly describes the origin of replication? Question 7. Match the following proteins with their role in DNA replication. Question 8. Match the following enzymes with their function in DNA replication. Question 9. Identify the leading strand and the lagging strand. Question 10. Which of the following Okazaki fragments was synthesized first?